So today we're covering blood. And so a lot of us already know a lot about blood. Everybody in here has experienced bleeding in one way or another. You cut your finger, you know what it looks like. You probably know what it tastes like because people tend to stick their fingers in their mouth if they get a cut. So that we already know a lot about blood. We also, from 2401, you hopefully remember that blood is a connective tissue. So remember the connective tissues, they have some characteristics. And one characteristic they have is they have more matrix than they do cells. And the matrix is what determines what the tissue can do. So if you look at, say, bone, the matrix is hard, and so it makes bone hard. If you look at cartilage, the matrix is this flexible, almost like bendy plastic material, and so that's what cartilage is like. So if you look at blood, blood has a liquid matrix, and because it's liquid, then it can flow. And so those we already know about, that, and that liquid matrix is called plasma. And then floating around in it are going to be cells. Now, right now, we're going to use the term cells, but they're not all truly cells. But right now, we're going to call them that. But they come in three different types. There are red blood cells, there are white blood cells, and there are platelets. And so we have this liquid matrix with these cells floating around in it. Well, if we put this blood in a centrifuge, when you centrifuge something, you spin it, and what happens is the heavier particles wind up down at the bottom. So this is the heavy stuff right here. And the lightest particles wind up at the top. Well, when you do that with blood, you wind up with three fractions. Three fractions. So the heaviest part is the red blood cells. And so the red blood cells are down here, and they're about here. They're packed in there together. The lightest part is the plasma. And so the plasma is the liquid portion of the blood, and it's right here. So there are no cells in it. Now that doesn't mean there's not other stuff in it. There's a lot of stuff in there, but no cells. So our red blood cells are down here. Our plasma's up here. Well, what's left is this little bitty layer in the middle, which is called the buffy coat. And the buffy coat is where the white blood cells and the platelets are. So you can see how many more red blood cells there are, white blood cells and platelets, just by looking at it. And you can see this with your naked eye. You don't need to look at it under a microscope. And so we can take this spun blood, we spin it in the centrifuge, and look at it. The total here is 100%. That's what we started with. Well, if we look at how much each fraction is of that 100%, you get an idea of how many cells there are. And so we can do sort of a quick and dirty evaluation to see if you have a normal amount of red blood cells just by looking at this. We can measure the percent of the red blood cells. It's called the percent packed cell volume, or it's called the hematocrit. So a hematocrit is something that's done to give us this sort of quick and dirty way of estimating how many red blood cells a person has. Make sense? So here's our blood, and it's got these three fractions. So there's the plasma, there's our red cells, and then here's the white cells and the platelets. Well, if you look at each one of these, they look different. So if we look at plasma, remember, is the liquid portion of your blood, and it's mostly water. It's about 92% water. but there's a lot of stuff in it. So it's not pure water. There's a lot of stuff in it. There are a lot of proteins in it. And if you do the PowerPoint on blood, it would talk about those different proteins. But it's about 7% are proteins, different types of proteins. Some of these have to do with things like keeping osmotic pressure normal. Some of them have to do with things like transporting hormones, or they may have to do with fighting off immune or pathogens. There's one in here that has to do with blood clotting, and that's called fibrinogen. And then there are also proteins that are actual hormones, so they're regulatory proteins. But that's not all that's in there either. There's a ton of ions in here. So if you look at the ions that are in here, there's all kinds of them. There's sodium and potassium and chloride and hydrogen ions and calcium and bicarbonate and all kinds of ions. Well, because they have a charge on them, we usually refer to them as electrolytes. So they are charged particles. They are electro. And there's going to be a bunch of these in here as well. Not only that, but we're going to have nutrients in here. Nutrients are things like glucose and amino acids and all sorts of things like that. Fatty acids, all kinds of nutrients, vitamins, stuff, anything that's nutritious or important for our health and welfare. There are also dissolved gases in here. So when we get to respiration, we're going to talk about two gases, one of them being oxygen and the other one being carbon dioxide. Well, a little bit of oxygen and even more carbon dioxide is transported, just dissolved in the plasma. And then there are waste products in here too, and the waste products are things like urea and ammonia and uric acid. Those are also transported in the plasma. So if you look at plasma, it's a very, very complicated liquid. It's got all this stuff in it. It's also very valuable. The reason it's so valuable is because you can give it to anyone and not worry about matching it. So it can be given to anyone in emergencies. And so you can take it with you in case of an emergency. You can just give it to anyone. Another good thing about it is you can freeze it. You can freeze it, and if you freeze it, you can store it. And it can be stored for up to 2 years. And so you can freeze this stuff. You can fly it all over the country or all over the world. You can give it to anybody without worrying about transfusion issues. And what it does is it increases the blood volume. And if you increase blood volume, you increase blood pressure. And so humans cannot live, no animals can live without blood pressure. And so if you in an accident and you're bleeding a lot, you lose blood pressure and you actually die from a loss of blood pressure. And so we can put that in there and bring blood pressure back up. And so it's a really, really important part of the blood. But this lab is mostly about the cells. And so let's look at the cells. So here is a red blood cell. And here is a white blood cell. And here's a platelet. Well, in reality, only white blood cells are actually cells. These others are not really and truly cells. A red blood cell has no nucleus. It also has no organelles. And so it can't reproduce, it can't do any of the things that normal cells can do. And so in reality, it's not a cell at all. It's basically just a little bag of something, a little bag of hemoglobin. And it floats around until eventually it wears out and breaks down. Platelets are also not cells. They're little tiny pieces of cells. They're just little bitty pieces of cells, and they last very long either. They only last about 5 to 7 days. So the only ones that are really and truly cells are white blood cells. And we're going to look at white blood cells first. So here are our white blood cells over here. Look at them. There are 5 different types of them, 5 types. And you can divide those 5 types up into 2 groups. One group, this group, if you look at them, look, they look granular or grainy. You see these little granules in here all over the place. See these granules in here all over the place and in here. So they're granulocytes. They have granules in their cytoplasm. But these, if you look at them, they don't have any granules in them. And so those are called agranulocytes. And if you put A in front of a word, granulocytes. Anyway, if you put A in front of a word, it means no or without. So we can immediately divide them up into these 2 groups. So let's look at these white blood cells. Now, white blood cells are technically called leukocytes. Now, most of the time, I just call them white blood cells. But leuko just means white. That's what it means. And cyte means cell, so white cells. They're not really white. They're actually sort of clear. So in order to see them, we have to stain them. And when we put the stain on them, some things will absorb stain. Well, first person who actually came up with a good way of differentiating these white blood cells was a guy named Wright. And he invented this stain, which is called Wright's stain. It's not very complicated. All he did was mix 2 stains together. He mixed a stain called eosin, which is bright, bright, bright red, and it's acidic. He mixed that stain with another stain called basic blue. So basic blue is just like it sounds. It's basic, not acidic, it's basic, and it's blue. So we put these 2 stains together into one stain, and that's white stain. And what happens when you put it on cells is that the cells will preferentially absorb more of one or the other. So some things turn red, other things turn blue. And what he was able to see was he could see these granules. And so he could separate them into these 2 types, granulocytes and agranulocytes. And then you can further separate the granulocytes into 3 types. And you can further separate the agranulocytes into 2 types. And so we wind up with 5 different types of white blood cells. Let's look at the granulocytes first. So again, we already know granulocytes are going to have granules in their cytoplasm. And you'll be able to see them under the microscope. And these granules are going to absorb those stains. So if you think about it, remember we got 2 stains. One of them is eosin, and eosin is bright, bright, bright red. And so anything that absorbs eosin is going to turn red. And then we have that other one, which is called basic blue, and it's a very, very dark blue. And so anything that's going to absorb it is going to turn this very, very dark blue color. Well, what would happen if you absorbed both stains? What do you get when you mix red and blue? You get purple. And so some things are going to absorb both and they're going to turn purple. So we have 3 different types of granulocytes. And look at their names. The name ends in the word phil. Phil means to love. So in other words, they love something. And what they love are those stains. So look, eosinophils love eosin. What color is eosin? Anybody remember? So they're going to absorb this stain. They're going to turn red, the granules. Look, basophils love basic blue. So when they absorb the stain, they're going to turn this dark blue. But neutrophils are neutral. In other words, they love both of them equally. And so they're going to turn this purple color. And so that's how we're going to differentiate them, or at least one way, by color. So let's look at these 3 types. Let's look at neutrophils first. So this is just a drawing of a neutrophil, but look, you can see these granules. And some of these granules will absorb the blue, some of them will absorb the red. And so it's going to give this a purplish look to it. But the only thing is these granules are very small. They're small granules, and they're actually hard to see. So if we had to depend on the granules, it would be a hard cell to identify. But fortunately, there's something unique about this cell. And that is, if you look at the nucleus, it's very weird looking. It has 3 to 5 lobes. Look at this nucleus. 1 lobe, 2 lobes, 3 lobes, maybe even 4. And they're connected by little things that look like strings. That is a very strange looking nucleus. Most nuclei that we think of are going to be round or some form of round, but these are not like that. And so the easiest way to recognize them is just to look at the nucleus. So if we look here, you can see those little granules in there. They're really hard to see because they're so small and indistinct. And that's that purplish color. Depending on how long they stay in the stain, it's a different color purple. But look at this nucleus. If you look at that nucleus, look, 1, 2, 3, 4 lobes. 1, 2, 3, 4 lobes. 1, 2, maybe 3 lobes. So neutrophils don't look like any other white blood cell. Neutrophils are the most common type of white blood cell. Most common. And so when you look at a blood smear, you see tons of these. These are bacteria killers. These are the ones that are most involved in fighting bacteria. And about 70% of all of your white blood cells are going to be neutrophils. So by far, they're the most common. So, more neutrophils. Can everybody recognize the neutrophils? Okay, let's look at another one. Let's look at eosinophils. So remember, phil means to love. Eosin means that red stain. Look at these red granules. They're very large and very easy to see. They're all over the place. So this cell is going to be bright red because of these red staining granules. Another thing is the nucleus here tends to be bilobed. Look at it, it looks like this. So it looks like a horseshoe. So 2 lobes. Now sometimes it can look like this. That's still 2 lobes. It's a bilobe nucleus. These are not very common. It's somewhere between 1 and 4% of the total are going to be eosinophils. And eosinophils are all about fighting off parasites. Things like fighting off worms or something like that. And so if your dog or a human or whatever were to get parasitic worms, this eosinophil count would go up. So here's what eosinophils look like. Look how red those granules are. Really easy to see. And then there's our bilobe nucleus. There's our bilobe nucleus. Now this is a little harder to see. It's just a staining issue, but that one is very easy to tell. Everybody pick out eosinophils? Let's look at basophils. So again, phil means to love. Baso refers to that basic blue. And here are these granules. Now this picture though doesn't have very many granules. Usually there's tons of them. So if we were to add some more granules in here, it would look like this. There's so many granules, a lot of times you can't even see the nucleus. It's obscured. That's how many granules there are. But if you can see it, it tends to be bilobed. This is the least common type of white blood cell. It's only about 0.5 to 1%. Very, very few of them. These are kind of unique. These granules that you see, some of these granules are histamine. And histamine is what increases the symptoms of inflammation. So inflammation is what happens when your tissues get infected or something, and they're going to swell up and they're going to turn red because more blood's going to come there, and they're going to feel hot to the touch, and they're also going to hurt. Histamine causes most of these symptoms. And so basophils are very much involved in inflammation, but they're also involved in allergies. So allergies is a type basically of inflammation. What happens with basophils is that they have a receptor on their surface where a particular type of antibody can attach. And so if you're allergic to something, you'll make these antibodies. And what they'll do, they're called IgE. What they'll do is they'll find a basophil and they fit in this little receptor and they'll hook up with it. And if you ever encounter that same thing that you're allergic to again, what's going to happen is, here's the thing you're allergic to. It's going to bind to the antibody, and it's going to cause this cell to release histamine. So it's going to degranulate. The granules are going to come out, and you're going to get all kinds of symptoms associated with allergies. So depending on where the allergy is, you might get a stuffy nose, you might sneeze, your tears might increase, your eyes might swell up, you get a lot of itching and coughing. All of that, or at least most of it, is caused by histamine. So if you think about what you do to relieve those symptoms, we take an antihistamine. Well, this other process still happens. So all of this happens. Our antigen and antibody, we're going to release the histamine, but histamine has been blocked by the antihistamine. It blocks histamine, and so histamine, even though it's floating around, has a smaller effect. So if we look at those, they look like this. So look at all those granules in there. There are tons of granules in there. And there's so many that it's hard to see the nucleus. Like I can pick out the nucleus looks like that. So it's bilobed. I might be able to pick out the nucleus. It looks like maybe that. It's bilobed. But the nucleus is obscured. by these granules. Now this one I can see, but over here I can't even tell where the nucleus is really. Everybody pick out a basophil? So Deli says the antibody works with the basophils to release histamine. So if you're allergic to something, we're going to make a particular type of antibody. We'll get to this when we get to the immune system, but it's called IgE. So IgE is the antibody that's made if you're allergic to something. So I'm allergic to cats. So if I'm around a cat, the dander from the cat is going to get in my nose and stuff, and my IgE is going to attach to it. IgE can attach to basophils. These cells right here, there's a little receptor that it fits in. And what happens is when this happens, then all these granules in here get released. And so this cell is going to release histamine. And it's the histamine that's going to produce the symptoms. And so what happens to me when I get around a cat is my eyes begin to itch like crazy and they swell up. No other symptoms except my eyes, but I'll get to the point where I really cannot even see. That's how they swell and they become so itchy and they tear like crazy. It's just my eyes, nothing else. Now that's not going to kill me, but it certainly is not pleasant. So I have friends with cats, and so if I go to their houses, this happens to me. And so if I know they're going to go there ahead of time, I'll take an antihistamine. And so when I get there, even though this whole process is going to happen, the histamine is blocked, and so I don't get as many of the symptoms. Does that answer your question, Nellie? Okay, so those are the granulocytes. Let's look at these agranulocytes. So remember, A in front of a word means no or without. So no granules. So when you look at these, you're not going to see those granules. You're going to see is a fairly clear cytoplasm. It's somewhat sort of like lilac color. It's clear and lilac colored. There are two types of these. So there are monocytes and there are lymphocytes. So let's look at these. Let's look at monocytes first. So monocytes are big cells. They're very big. This is not a monocyte. This is a monocyte. The reason I put this picture is in here so you could compare a red blood cell with a monocyte. Monocytes are 3 to 4 times bigger than a red blood cell. So when you see one, they're pretty easy to pick out. They also don't tend to be round like that one is. They have sort of a shape like this. And then they have a bilobe nucleus, but no granules. So monocytes turn into something called macrophages. Macro means big, and phage means to eat. So they're big eaters. Big eaters. In fact, that's what they do. They go around eating things that are not supposed to be there. So there are major phagocytes. They're very, very phagocytic. Now, monocytes, it's about 12% of the total. So there's quite a few monocytes. So here are monocytes. So look, these are red blood cells, and look how big this cell is. Again, it's 3 to 4 times bigger than a red blood cell. And if you look in there, you don't see any granules. It's just clear, pale blue, lilac colored cytoplasm, and then the nucleus is bilobed. So remember, a bilobe nucleus can look like a horseshoe. It can also sort of look like a dumbbell. So those are monocytes. Everybody recognize monocytes? Again, look how big this thing is compared to that. They're huge cells. Let's look at lymphocytes. So lymphocytes are the second most common type. It's about 20 to 25% of your white blood cells are going to be lymphocytes. Look at their name though, lymphocytes. In fact, most of these cells are not in blood. We see them in blood, but most of them are not in blood. They're in lymph. And so in lymph, which is another fluid that our bodies have, there's millions and millions and millions of them. And so since they're mostly in lymph, they're called lymphocytes. Well, these are the smallest of the white blood cells. So they're going to be about the same size as a red blood cell. And the easiest way to remember these is they're a round cell with a round nucleus. They're the only ones that are like that, a round cell with a round nucleus. Now in this picture, you can see a lot of cytoplasm, but usually there's only a little bit of cytoplasm. The nucleus takes up almost the whole cell. So there's only going to be a little bit of cytoplasm that you can see. So if you look at lymphocytes, here they are. So look, round cell, round nucleus, and only a little bit of cytoplasm. Round cell, round nucleus, and only a little bit of cytoplasm. Make sense? Can everybody recognize lymphocytes? So remember, they come in different percentages. The most common one was the neutrophil, and the least common one was the basophil. So if you want to remember these in order, there's a little mnemonic that can help you. And it's never let monkeys eat bananas. Never let monkeys eat bananas. And that's from most common to least common. Never let monkeys eat bananas. Of course, they're not going to like you very much if you don't let them eat bananas, but that's the mnemonic. So those are the white blood cells. Any questions about them? Okay, well, let's look at red blood cells. So just like white blood cells were called leukocytes, red blood cells are called erythrocytes. Now, I don't use this word very much either. I just say red blood cells. But remember, red blood cells are not truly cells. They don't have a nucleus. Now, they start out with a nucleus, but as they develop, they actually spit the nucleus out. They eject the nucleus along with all of the organelles. But what happens when you eject the nucleus is it changes the shape of the cell. So it's not really round anymore. If you look at it from the side, shaped like this, it's sunk in on both sides. And so the shape is called a biconcave disc because it's concave on both sides, a biconcave disc. In other words, there's no nucleus, no organelles. It's really just a little sac. It's just a little sac, a little bag. And it's full of something called hemoglobin. And hemoglobin is what we use to transport gases. And so that's what their job is, to transport gases. So when you look at red blood cells, there's tons of them. But look, you can see this sunken in area here. In some slides, some pictures, it's going to look like a hole. Like these look like donuts. But it's not a hole, it's just a very thin area. They're biconcave discs. You see them here. Here's an even better picture where you can see them. Well, we have a ton of red blood cells. We have about, on the average, about 5 million of them in every milliliter of blood. So there's a ton of them. So when you look at a blood slide, you're going to see red blood cell after red blood cell after red blood cell. There's just tons of them. Any questions about red blood cells so far? Well, red blood cells, on their surface, they have antigens. Well, an antigen in this case is just a protein sticking out. So here's this protein. And these proteins can create problems. So we've known for a really, really long time that if someone was bleeding to death, the best way to save their life was to give them more blood. In fact, even back in the Middle Ages, transfusions were being performed. Now, they did it directly from one person to another, so they'd take one person and they'd take another one and they'd pump the blood straight out of this person into this one. They'd connect the two. Well, that sounds like a good thing, but actually, you had a 50-50 chance of dying from the blood transfusion. So in other words, 50% of the time, the blood went in there, this guy would live and he would hop up and he would feel much better. But 50% of the time, laying there on the table, he would just die. And nobody really understood what was going on. The first person to check this out was a doctor, and he was working in yellow fever, actually. But he noticed that when people got transfusions, a lot of them died. And so he wanted to find out why. And so what he did was he took blood from all of his patients and also all of his staff. And he put them in tubes like this, and he labeled them and so on. He kept really good records. But here's just all this blood, just blood. And so here's the blood in here, and here's the blood in here. And then what he did was he took a little bit of blood out of each one and he would mix it with the others. So in other words, he used an eyedropper kind of thing and took blood out of here, and then he would put it in here, and then in here, and then in here. And then he would watch what would happen. Well, about 50% of the time, nothing happened. The blood just looked like blood. Nothing happened, everything was fine. But about 50% of the time, what happened here was the blood would clump up. It would form these big, thick clumps. It would agglutinate. Now, it's not a clot, it's a clump. It would agglutinate. But again, 50% of the time, nothing happened. And 50% of the time, it would clump up like crazy. And so what he figured out was that when it was normal, this person would live. But if it agglutinated, this person would die. Well, he still didn't know why it was happening. But he was able to, by mixing all of these blood, so he'd take now out of here and put it in here, and down here and here, and again, watch what happened. Take it out of this one and put it in here. He would watch what happened. He found out, or figured out, that there were different types of blood. And he saw that there's different types. He found 4 types, and he named one of them A, and he named one of them B. He named one of them AB because of the way it reacted, and then he named the last one 0 because there weren't any reactions. Well, 0 over time has been shortened to O. And so what we wind up with are 4 blood types. So we have type A, type B, type AB, and then that type 0, which is now called O. And so if you look here, you can see that on the surface of these cells are these different proteins. So on A, there's this protein he named A. There's this B, there's this protein he named B. On AB, there's the protein A and the protein B. And then type 0 did not have either one. It didn't have any. It had neither. And then, of course, that became O. I like 0 better because it tells you something about what's going on. But that still did not explain why this was happening, this clumping that was happening. It was much later when someone else figured out that what was going on is that in our guts, in our large intestine, we have billions of bacteria in them. And those bacteria, if they stay in the gut, they can't do anything to hurt us. But if they get into our bloodstream, they can cause disease. And so to prevent that from happening, we build antibodies against that particular bacterium to prevent or to get rid of the disease. So this is an antibody that we're going to build. But antibody attacks things that are not us. They attack what's called non-self. It's important that they attack non-self because that protects us, but it's also important that they do not attack us. They do not attack self. Does that make sense to everyone? So does everybody understand the difference between self and non-self? So self is anything that's part of you, any protein, any cell, anything that's part of you, that's self. Everything else is non-self. So it's important that we attack non-self, but it's more important even that we do not attack self. So let's go back and look at this again. Here we are, and we've got these blood cells. And these blood cells have these proteins on them. So let's say that this person is type A. So what they have on their red cells are these antigens called A. So this person's type A. Well, they also, though, have in their large intestine these bacteria. Well, guess what? There are bacteria in here that also have A on them. And there are bacteria in here that have B on them. In every person, we have these bacteria with A's and B's. Well, look, I am going to want to protect myself against these bacteria. I'm going to make antibodies. But look at this. This bacteria has A on it. It's the same thing that's on my red cells, this exact same protein. If I make an antibody that can attack this, that same antibody can attack this. Because what is A? Is A self or non-self? It's self, and I cannot attack self. So I cannot make that antibody. But look at this. Here's another bacteria here, and this one has B on it. Well, I don't have any B's on my red cells. So what is B to me? Is it self or non-self? It's non-self. And so what I'm going to do is ahead of time, before anything, because of these bacteria, I'm going to make a bunch of anti-B antibodies. So if I look at this person's blood, not only do they have red blood cells with type A on it, floating around in their plasma, they have anti-B antibodies. And if that ever comes in contact with the B cell, it's going to attack it and kill it. So that's A. But let's look at another one. What if I'm type B? So I'm type B. What is B to me? Self or non-self? It's self. But what is A to me? Self or non-self? It's non-self. So look, same situation. I've got the gut. I've got the bacteria in here. Those bacteria have A on them and they have B on them. Can I make an antibody against the A? Yes. Can I make an antibody against the B? Can I? No. So what's going to happen is this person's going to have B on their red cells and they're going to have anti-A floating around in their blood. If that anti-A were to ever come in contact with a blood cell that had A on it, it would attack it. Let's go back one more. Look at this person. That's wrong color. Look at this person. Here's their blood type. What is this person's blood type? It's AB. So for this person, what is A? Is it self or non-self? It's self. What is B? Self or non-self? It's also self. So can I, even though there's bacteria in the gut with A on it and with B on it, can I make any anti-A antibodies? No. Can I make any anti-B antibodies? No. Which means that in this person's blood, I'm not going to find either one of them. I'm not going to have either anti-A or anti-B, neither one. Does it make sense? And then we have one more. So look at this person's blood type. Here's their red cells. What is this person's blood type? They're type O, that's right. So look at this though. They still have this gut thing. There's still bacteria in here with A and with B. What is A to this person? Is it self or non-self? It's non-self. What is B to this person? It's also non-self. So when this person sees these bacteria, when their body sees it, can I make anti-A antibodies? I can. Can I make anti-B antibodies? I can. So floating around in this person's blood are going to be both anti-A and anti-B. And if I ever encounter A, I'll destroy it. If I ever encounter B, I'll destroy it. And so when you look at this person's blood types, look, this is what we see. This person's type A. They have A on their red cells, and they have anti-B in their plasma. This person's type B. They have B on their red cells, and they have anti-A in their plasma. This person has both A and B, and so they can't have anti-A, and they can't have anti-B antibodies. They have neither one. But this person is type O. So that means there's no A's, no B's on their red cells. They're going to make both anti-A and anti-B antibodies. And then what's going to happen is if I mix this into this blood, that's going to attack it. If I mix this into this blood, that's going to attack it. If I mix this into the blood, it's going to be attacked by both. And this is what's going to happen. Make sense? So, if you have the if you have the antigen A, you're type A. If you have the antigen B, you're type B. If you have both of them, you're AB. If you have neither, you're O. So New says O type have a better immune system. No, not necessarily. I mean, against A and B, yes, but not against anything else. But you're right about A and B. They can't attack, or they can attack A and B, not like the others can. But anyway, and then we have these antibodies, anti-A and anti-B. And so this is what's going to happen. Look, this person is type A. So that's what their blood looks like, and that's what their plasma looks like. But they've had some sort of injury. So we're going to give them a transfusion. But look at what the transfusion is. It's got B on it. That is specifically designed to attack that. So as soon as this gets in here, that's what's going to happen. These antibodies are going to attack and destroy this blood. It's going to agglutinate, and then we're going to get destruction. We're going to get hemolysis. And this person will probably die. Any questions? Does everybody understand type A, type B, type AB, type O? Well, that's weird. But anyway, type A, B, and type O, everybody understand it? Any questions? So we want to be able to give people transfusions. So we're going to have to know what their blood type is, and we're going to have to know what the blood type of the donor is. So we have to have some sort of test to figure out the blood type. Well, guess what? We're going to use the same process that we have here. We're going to use antibodies to figure out what will attack. Look, that's specific against that. That is specific against that. So if we could isolate these antibodies, we could use them to see if that was on a cell or not. If we could isolate these antibodies, the only thing this will attack is that. The only thing this will attack is that. And that's exactly what they do. So what they do is they take something like a rabbit. Oh, I don't know if I can draw a rabbit with a mouse, but anyway, they take a rabbit and they inject it. And let's say they inject it with type A blood or type A antigen. Well, rabbits don't have type A. And so what's going to happen is they're going to build anti-A antibodies. So we're going to take and we're going to get the blood out of that rabbit. We're going to separate these out, and we're just going to put them in a vial, in a bottle. And so this bottle is anti-A. And what is the only thing anti-A will attack? The only thing it will attack is what? It attacks A. So if we get agglutination, we're going to know that A is present. We're going to do the same thing with a different rabbit, and we're going to put type B in there for this rabbit. So when we put type B in, we're going to get anti-B antibodies, and anti-B antibodies only attack B. And again, we're going to put them in a bottle. And so we're going to have these bottles of anti-A and anti-B. So anti-A only attacks A. Anti-B only attacks B. What we're going to do is we're going to take a blood sample. So these are 4 different people. This is all the same blood from number 1. This is blood from number 2. This is blood from number 3. And then what we're going to do is we're going to combine it with anti-A and anti-B just out of our little vials. So look, here's a blood sample with neither one, so you can just see what the blood looks like. And then the next drop of blood has anti-A mixed with it. And the next drop of blood has anti-B mixed with it. And it's that way all the way down. And so we can tell what this person's blood type is by looking at this. So see if you can figure out what this person's blood type is. See if you can do it. Figure it out and tell me. Are they A or B or AB or O? So Soon says A. That's exactly right. This person's A because look, we got clumping in here. It was mixed with anti-A, and the anti-A only attacks A, which means that A is present. But look, there's no clump in here, no clumping. Anti-B only attacks B. And if there's nothing to attack, nothing's going to happen. So there's no B. Make sense? What is this person's blood type? A, B, AB, or O? This one's B. That's exactly right. So same thing. Here's anti-B, only attacks B. We got clumping. No clumping here, so there's no A. What about this person's blood type? It's AB, exactly, because look, we got clumping here, and we got clumping here. So A is present and B is present. And then what is the last one? It's O, because there's no A and there's no B. So it has to be O. And that's exactly right. Does it make sense to everyone? Well, there's one more thing that we can find on red cells. Actually, there's a bunch more things, but there's one more thing that creates problems. And this is called RH. RH stands for rhesus monkey is what it stands for, because that's where it was first discovered. But humans have it too. So this RH is another antigen. It has absolutely nothing to do with A, B, and O. It's completely separate. But look, this person has RH. This person does not. So this person is said to be RH positive. This person does not, so we say they're RH negative. But it has nothing to do with the A and B. So this person have an A on here. So if I look at this person, they're type A, RH positive. Or if I want to just shorten that, I just say A positive. This person is also type A, but they don't have any RHs on them. So this person's blood type is type A, RH negative. Or if I want to shorten that, I would just say they're A negative. And so we can have A positive, A negative, B positive, B negative, AB positive, and AB negative, and O positive, and O negative. So everybody see how I got all those different blood types? So if I pick that person, their blood type, their blood cells would have B on it, but no RHs. But if I pick this person, they would have both A's and B's and RHs. Does it make sense to everyone? So look, this is also important. So I'm going to do the same thing I did before. I'm going to find a rabbit, but this time I'm going to inject the rabbit with RH. So there's my little rabbit, not a very good one, but anyway, I'm going to inject it with RH. And what I'm going to get is anti-RH, and I'm going to put it in a bottle. And then what I'm going to do with it is I'm going to mix it with the blood and see if it clumps up. So look, this person's blood, is it positive or negative? Positive. What about this one? Positive. What about this one? Positive. And that one? Negative. So what I want to do is do the whole thing all together. So positive, 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 negative. So now, when I want to do a blood type, I do all three. So if you look at this person's blood, see if you can tell me what this person's blood type is. A positive. Exactly, Soon. What about this one? B positive. What about this one? AB positive. What about this one? This is AB positive. What about this one? O negative. So can everybody figure out a blood type? So you might get something like this on the lab test. Now, of course, it's going to be all mixed up. It won't be this one. But it doesn't matter as long as you know which. All of these had anti-A mixed with them. All of these had anti-B mixed with them. And all of these had anti-RH mixed with them. So you can figure out the blood type. Can everybody do that? So what we need to know then is the blood type of the donor, and we need to know the blood type of the recipient so we can match the two up. And remember, you can't just give willy-nilly, because if you give the wrong blood type, remember this is going to happen. We're going to get this happening, and hemolysis, and probably death. So we have to be really, really careful about mixing these different blood types. And so if we want to look and see who can get what, we use this. So this is a chart that shows who can give to whom. So look, this right here is the donor's blood. That's the donor. And this right here is the recipient's blood. Now we already know A can give to A because that's the same. B can give to B, that's the same. We already know O can give to O. And AB can give to AB because that's the same as well. But some of these others can also give. And the way you figure this out, remember, is you remember what the antibodies are. We cannot give something that there's an antibody against. And so when we look at this, these are the ones that each one can get. So if it's AB negative, look, they can get from A negative. That's fine. They can get from B negative. That's fine. They can get from AB negative. That's fine. And then they can also get from O negative. So negatives can give to positives, but positives cannot give to negatives. And then we can't cross-match the other things as well. But if we look at this, look, there's one blood type here who can give to everybody. Every single recipient can get this blood type without any kind of problem. That's O negative. So O negative is sometimes called the universal donor because there's not any antigens present. There's nothing on that O to attack. There's no A's to attack. There's no bees to attack. There's nothing to attack. And so O negative can be given to anybody. And so it's a very, very valuable blood. And so blood banks try to keep this on hand very, very much, because this is what's used in emergency rooms and places like that when they don't have time for blood typing. And so if you happen to have type O negative blood, you're going to be a very, very popular person with a blood bank. They're going to love you. And they're going to love you so much, they're going to drive you crazy asking for your blood. There's also, if you look on here, one blood type that can get blood from anybody. Look at that. Anybody can give this person blood without any risk of rejection. And that's type AB positive. And so sometimes AB positive is called the universal acceptor or universal recipient. But let's go back and look at this. Who can AB positive give to? They can only give to AB positive. So if you're AB positive blood, you are really, really lucky, because you can get blood from anybody, but you can only give blood to AB positive. So nobody cares about your blood. So you have a universal donor and a universal recipient. Any questions? So what I'd like you to do, and I'll send this out, is there's a place where you can go and do what's called the blood typing game. And it's kind of fun. It's by Nobel, like the Nobel Prize. Anyway, I'll send out the link, and you can go there. And what it is, you're going to pretend you're the doctor and you have patients, and they're going to come in and they need blood, and you're going to have to figure out the blood that they can get from what you're allowed from the blood bank. And you're either going to save the patient or you're going to kill the patient. The only two choices. But it's kind of fun to do. So I'll send that out. Okay, so those are red cells. Let's look at platelets. So platelets are technically called thrombocytes, but I don't know anybody who calls them that. They're just called platelets. Remember, they're not cells. They're just little tiny pieces of cells. So there's this giant cell in the bone marrow, and what happens is it sheds cytoplasm. And so these little pieces that come off of it, those are platelets. When you look at them, they have granules in them. They're not alive, so they don't last very long. They only last 5 to 7 days, and then they're removed from circulation. They're removed by the white blood cells in the spleen. And then new ones are produced and released. Their whole job is about blood clotting. So if we look at them, they look like this. They're these little bitty pieces of cells. They don't travel singular usually. They travel in bunches. And I don't know how many's in that bunch, but it's probably 100 or more. There's one by itself. You can see how small they are, but again, they mostly travel together. Any questions about platelets? Okay, so the rest of this is just some practice, and I'm not going to really go over. Well, we can go over it. But rather than me asking you, though, I'll just tell you. But look, there's 3 different types of white blood cells here. So that's a neutrophil. These are monocytes. And then if you come over here, these are neutrophils. In fact, all of these are neutrophils. Those are lymphocytes. There's an eosinophil. There's a basophil, there's a monocyte, and there's a neutrophil. There's another eosinophil. There's a neutrophil, a lymphocyte. That's a basophil. The more you do it, the easier it gets. So I would suggest that you practice because you will be asked to identify them in the lab test. Now I try to pick the ones that are the best ones. I'm not going to try to trick. So some weird looking cell that doesn't look normal, I probably would not pick that. Even though it's a lymphocyte, it doesn't look like a typical lymphocyte. I sure wouldn't pick something like that or like that. And that one's messed up, so I wouldn't pick that. But these are really nice neutrophils. So don't worry and think. And this one is kind of hard to tell because look, that's not red, that's not blue. This is actually an eosinophil, but it's a very bad stain. So I wouldn't pick that either to ask you. I'll try to pick something that's fairly obvious. Any questions? Why can AB positive receive any blood? No. Negatives can give to positives. So negatives can give to positives and positives can give positives. So remember this person's AB positive, which means they can get all of the negatives and they can get all of the positives. That means they can get any blood. You can't do it in reverse though. Positive cannot give to negative. That's why AB positive could not give to AB negative. Remember, AB positive can only give to AB positive. They cannot do that. They can't give to negatives. Positives cannot give to negatives. Does that answer your question, Deli? Anybody else have a question over any of this? It doesn't have to be the cells. But again, here's a nice chart to show you who can give to whom. So make sure you understand why each of these can do each of these. But look, negative can give to positive, but positive cannot give to negative. Oops, positive cannot give to negative. Anybody else have a question? And O blood cannot receive anyone except O. That's exactly right. But if you're O negative, you can only get blood from O negative. That's it. But if you're O positive, you can get blood from O negative and O positive. Because negatives can give to positives. See, if this is the patient right here, if you're O positive, look, you can get O negative blood and O positive blood. The only one that can only get its own blood type is this one. So if you're O negative, you know, you've got double trouble. One is all the blood banks want your blood. They all want your blood. But the only blood you can get from when you get to the hospital is this same type. It has to be, must be O negative. That's it. Now there is a company that has developed a process to remove the A, and the B, and the RH. So look, if you're A and you take the A's off, it basically become O. And if you're B and you take the B's off, you basically become O. And if you're AB and you take both of them off, you basically become O. And if you take off the RHs, that means that every blood is O negative, which means if you use this process, it's a process of enzymes. If you use this process, you can create O negative blood from any blood type. So eventually, maybe this won't even be an issue. Any other questions? Okay, well, we're going to stop there. Thank you for coming. And if you have any questions, remember you can always email me. I'll email you back within a day. If you haven't taken all the quizzes and all the exams, you need to go ahead and do that. Try to stay caught up. There's going to be a new quiz today over this, and it'll, of course, be an announcement. But anyway, thank you for coming.